response that we see to a salmonella protein curly. So the disease that we're interested in here is systemic lupus erythematosus. This is a systemic autoimmune disease. It is characterized by anti-nuclear antibodies against things like DNA and chromatin. And it involves this widespread inflammation and can lead to tissue and organ damage. One of the organs that can be affected by lupus is the kidneys. So you can end up with lupus nephritis in about 40% of patients. An infection is the leading cause of morbidity and mortality in SLE and is associated with flares. Particularly common infections are E. coli, staph, and strep. Salmonella, non-typhoidal salmonella, so the kind that causes gastroenteritis, tends to be more aggressive and invasive in these patients as well. And so what our lab is looking at is what are potential mechanisms that are triggering these flares or triggering autoimmune disease after these infections. So our kind of candidate protein for what might be triggering these responses is curly. It is a bacterial amyloid that's present in Salmonella and E. coli biofilms. Biofilms are multicellular aggregates of bacteria surrounded by an extracellular matrix that provides them protection, resistance to stress. And this matrix for Salmonella and E. coli is primarily composed of cellulose, extracellular DNA, and the bacterial amyloid curly. And I'll also point out a lot of human infections involve biofilms. And about 40% of bacterial species can produce amyloid similar to curly. So the things that we're learning about curly may also apply in other infections. So the autoimmune response that we usually see in wild-type mice when they are either infected with salmonella that is able to produce curly or injected with purified curly from biofilms is the wild-type infection. You get these anti-double-stranded DNA autoantibodies. Those are like what you can see in SLE patients. And they also get this low-level knee inflammation, which might be more characteristic of a different autoimmune disease that can happen after these infections, reactive arthritis. And you see that in our wild-type salmonella infection, but not in the curly deficient mutant salmonella. And then the same thing as if you inject just purified curly minus an infection. So our model that we are working on is that when you have a biofilm of just like a commensal or like a non-invasive sort of bacteria, it can have curly inside the lumen of the gut. But as long as it stays inside of the lumen and you have like a healthy gut epithelial barrier, it actually is not producing this autoimmune response. But when you have an invasive infection, or where you can get this leakage through the gut barrier, you have this inflammation and damage, that is what can produce this autoimmune response. And so what my dissertation project is working on is how does this curly-induced autoimmunity interact with other like risk factors like genetics and other kind of predisposition. And this part of my project is looking at lupus. So to start out with here, we sent serum from either Black 6, oh, it's cut off on the top there, but Black 6 wild type mice or NZBW mice, which are a lupus model of, so they get the same sort of symptoms that humans will get of lupus. They get those autoantibodies. However, these were treated before the onset of disease usually. So these are not at the point where they are getting autoantibodies on their own. This is well before where they've been injected with curly. And we send these for a multiplex assay to look at a bunch of different autoantibodies. So one of the ones that came up in this multiplex assay of a bunch of different antigens that happen in lupus, in both the Black 6 and the NZBW models, we get this anti-C1q autoantibody response. So this is interesting because C1q is the kind of initiator of the classical pathway of complement. It's an innate immune response. And these anti-C1q antibodies are especially interesting because they have a very high association with lupus nephritis, so the subset of patients that have kind of kidney sequelae in lupus. And so one of the things that can happen with lupus nephritis is you get this buildup of immune complexes. So these immune complexes are composed of complement proteins like C1q or C3, as well as the autoantibodies against C1q or even autoantibodies against nuclear antigens. And then this can also be involved in other like inflammatory responses and eventually tissue damage in the kidneys. So first, we confirmed that we actually did see these anti-C1q antibodies in black 6 mice that were either IP injected with curly, just purified curly, or infected with salmonella. So these are black 6 mice. We had to use an attenuated strain of salmonella, but even so, we did see an anti-C1q response with the infection. And this is just in wild type mice. So of course, since this is relevant in lupus, we wanted to also look again in these NZBW mice. We looked at a couple different time points here. So one set was pre-onset, like weeks before they should actually be getting any disease. Early disease, which is kind of the typical onset is about 23, 24-ish weeks in our facility. So they're kind of getting disease in this set. And then late disease, which is weeks after the onset of disease. And these were IP injected with Carly. And so what we see with these anti-double-stranded DNA responses, which we usually see with Curly, in the pre-onset, you do see this increase in anti-double-stranded DNA. But in the early disease, as they're kind of already getting some autoantibodies because of the model itself, we don't see very much difference with Curly. There's a little bit of an increase, but it's not hugely significant. And then the late disease all have extremely high levels of this anti-double-stranded DNA, so we didn't see any difference with the Curly treatment. And so then moving on to look at these anti-C1q levels in both the pre-onset and early disease here, we can see an increase with the Curly both in the pre-onset set as well as the early disease set. But the early disease, so with age, they are seeing a higher level of these anti-C1q even with the treatment. So treatment plus age has even higher levels. The late disease already have some level of these anti-C1q that are developing just because of age, which is already known. And there's a little bit of a difference here with the curly group. So overall, it's a little bit elevated, but it's a little less of a dramatic difference in this late disease group since they're already getting some of these autoantibodies on their own. And so because of that huge variability, basically, that we're seeing in these NCBW mice, because of how they can be a little bit different in when their onset is, since it's so late, MRL LPR mice are a different lupus model, and these are a very kind of compressed timeline. So they start getting disease around 12 weeks old, and they're very, very rapid, very severe disease in this model. So the timeline here with, again, curly injections is kind of analogous to the early disease. So they're kind of getting onset of the disease as we're undergoing treatment here. And we saw a significant difference here with our curly treatment, which was a much higher level of these anti-C1q antibodies with curly treatment. And so, like I said earlier, immune complexes in the glomeruli are one of the things that are associated with lupus nephritis. And you can stain for these. So these are the glomeruli inside of the kidneys. You can stain for IgG, C3, so those complement proteins and antibodies. Those were not really any different between our curly treated and PBS treated. But then when we looked at C1q, what was really interesting is our curly group, which also had those significantly higher levels of anti-C1q antibodies, also had these much higher levels of C1q deposited in the immune complexes. And so the next steps that we want to look at, obviously, here would be more to see if we can see any other signs besides the immune complexes of pathogenesis inside of the glomeruli of these kidneys, both in our NZBW mice as well as the MRL-LPR mice. So these have all been processed and they've been sent to a collaborator at the University of Massachusetts, Dr. Caricchio, who is going to score the pathology for us and see if we see any signs of glomerular cell proliferation, leukocyte infiltration, tissue damage, to see if there's any other association with this anti-C1q, this higher response. And so in summary here, we have our genetic models of lupus, NZBW or MRL-LPR. With age, they're going to develop these anti-double-stranded DNA and these anti-C1q antibodies. But with curly treatment, and in this case, we were just doing IP injections, so not even looking at infection in this case. With curly treatment, you can accelerate the induction of these anti-double-stranded DNA in these models, although if they are already having high levels, it doesn't matter so much that they get the curly. But with the anti-C1q antibodies, it seems like despite them already getting an elevated level just from genetics, curly is like further increasing this anti-C1q response. It is also associated with higher levels of this anti-C1q in the immune complexes. So we are going to look for whether there's also other signs of lupus nephritis here, if that elevated C1q in the immune complexes has higher tissue damage or something like that. And one of the thoughts about why would curly even cause an anti-C1q autoantibody response has to do with curly's binding. So we already know from previous work that curly binds to extracellular DNA. And that's thought to be one of the reasons why we get this anti-double-stranded DNA response, almost like curly is like presenting the DNA in an immunogenic context, almost acting like an adjuvant even. And we have other work that shows that curly does bind to C1q as well. So it could be a similar thing that might be happening here. So that's something we're going to look at in the future. And I want to acknowledge everybody in our lab, and I mentioned a few of our collaborators as well who helped with this project. And I can take any questions.
I was curious, that was a very nice talk, by the way. Kind of answered my one question at the end, which was great. But I'm kind of curious, are there salmonella strains that have like variants in curly that have different induction of this at an anti-C1q response? That might be useful to think about.
Yeah, that would be interesting to see. So we haven't looked too much at different strains of salmonella. We know that it is able to be induced. So I showed already the one attenuated strain, that's SpyB mutant. So it doesn't need the SpyB is a type 3 secretion system for intracellular survival of the salmonella. So it doesn't seem to need that to induce the anti-C1q response. But beyond that, yeah, we're not sure. It would have to be a strain that has curly. But beyond that, yeah, it would be interesting to look at if there's like differences in the curly, then is there also differences in the anti-C1q? Yeah, we're not sure.
Cool.
Great talk, Caitlin. Really fascinating. So just curious, so I thought it was so interesting that you saw the anti-C1q antibodies in the B6 mouse strain also, because I always saw that those antibodies as being associated with like that background of, the mouse strain that's prone to lupus, right? The NZV cross XW strain. And so I noticed in your data, you saw that LA antigen, right? Also, there's increased antibodies in the NZW mice. Do you see that also in the B6 mice? Have you looked at that?
So that LA antigen, so that was one of the ones also in the Black 6 mice top graph, and we didn't see it in that. I mean, this was just one set of experiments, just with the curly injections. But we are actually thinking of looking at that in the future because I didn't show it here, but the MRL LPR mice that we treated with curly in this experiment, they had like skin lesions over top of the lacrimal glands, which is the anti-LA antigen is associated with Sjogren's syndrome, which has like inflammation in the lacrimal gland. So there might actually be something going on there with the like MRL as well. Well, we're going to look at it in the future. I'm not sure. So we didn't see that in the Black 6 so far.
Oh, okay. No. And I guess it just makes me kind of wonder, like I guess curly just seems to be like helping to like break tolerance to, right, like auto antigens. And and maybe your lab's done it. I don't know. I guess have you ever done mass spec with the curly to see what other host proteins are associated with it, and if there's like antibodies against all of those, with the universe of antibodies?
We have not done that specifically. No, done like a full just like panel of anything that is associated with curly. That would be really interesting to do though, to see if we see a similar sort of like just whatever is bound to curly becomes more of like an antigen. Yeah, we haven't done that yet, but that would be interesting.
Cool. Thanks.
Thanks.
Thank you so much.
Thanks.
Our next speaker will be Abigail Onafor.
All right, so today I'm going to be sharing my research titled Type 1 Interferons Disrupt Epithelial Cell Repair and Tight Junction Integrity in the Developing Lung, or in the flu-infected neonate, sorry. So first I wanted to start off on why we perform this research in the first place. So respiratory viral infections are the #1 cause of childhood hospitalizations in the United States. And premature infants are disproportionately affected because of their underdeveloped lungs, but also their developing immune responses. And while there is a flu vaccine, infants under the age of 6 months are not eligible to receive this. So there is a critical need to develop antiviral therapeutics that will target this age group. And in order to develop proper antiviral therapeutics, we must use age-appropriate models. So neonatal responses cannot be inferred through the use of adult models because at birth, neonates are rapidly developing their lung function, but like I said, also their immune status. And in humans, this rapid lung development occurs in the 3rd trimester and then continues into childhood. Mice also undergo this rapid lung development. However, theirs is restricted to the first 2 weeks post-birth. So a neonatal mouse is defined anywhere from 1 day old to 14 days old. And in our lab, we're focused on the high-risk preterm infant population. So we have developed a 3-day-old murine neonatal model of influenza A infection. So we infect our neonates on the 3rd day of life intranasally with the H1N1 strain of the flu. And at this point, the immune system and lung function represents that of a late preterm human infant, which is anywhere from 34 to 36 weeks gestation. Now, when we infect our neonates with an adult sublethal dose, weight adjusted on a per gram basis, you can see that all the neonates, which are shown by the solid black line, let me get a point. Right here, all succumb to infection by 8 days post-infection. Even when we reduce the dose, so now the neonates are receiving 4 times less virus on a per gram basis, we still see significant mortality during the first week post-infection. When we perform real-time PCR to look at the viral loads in the lungs during the first week of infection, what we find is that despite the fact that the neonates are receiving 4 times less virus on a per gram basis, they have significantly higher viral loads at 1 and 3 days post-infection compared to the adults, indicating that there is a delay or a defect in the neonatal immune response to recognize and control the virus. Now, one of the important innate immune responses to viral infections are type 1 interferons. So type 1 interferons are produced in response to influenza infection, and they can initiate antiviral gene transcription, they can activate immune cells, and their ultimate goal is to control the virus. Now, in adults, when you take out type 1 interferon signaling, so these are referred to as IFNAR mice because they have a defect in their type 1 interferon receptor. When you infect IFNAR adult mice and compare their weight loss to wild-type mice, you see that the IFNAR adults lose significantly more weight, which is a marker of morbidity, indicating that type 1 interferon signaling is required in adults to protect against flu infection. So we hypothesized then that the increased neonatal mortality we observe is driven by a defect in their type 1 interferon response. So we infected wild-type neonates and IFNAR neonates and tracked them for survival. Initially, we hypothesized that if you take out a key antiviral signaling molecule from an already vulnerable population, we would further exacerbate mortality. However, we actually found the opposite. We find that the IFNAR neonates now have 75% survival compared to the 20% in our wild-type controls. And that is because type 1 interferons require balance. So I talked about the protective effects of type 1 interferons, but unfortunately, there are also pathogenic effects, including cell death, inflammation, and reactive oxygen species production. And all of these are associated with severe influenza infection. And our lab has previously investigated the role of reactive oxygen species, and we found that there was type 1 interferon-driven ROS production from a subset of epithelial cells called type 2 epithelial cells. So we wanted to investigate this further. And you might be asking why? Why are type 2 epithelial cells so important? Well, firstly, they're located in the alveoli, which is the site of gas exchange in the lung. And they're also the target of highly pathogenic flu strains. And in the lung, they produce surfactants, which both facilitate oxygen exchange, but also modulate immune responses to different pathogens. And importantly, these cells are stem cell-like, so they can divide and replenish themselves, but they can also differentiate into these type 1 epithelial cells, which are the main cell subset responsible for gas exchange. So we hypothesized that type 1 interferon signaling within these type 2 epithelial cells contributes to neonatal flu pathogenesis. So to answer the question of how age and interferon signaling impact flu pathogenesis in type 2 epithelial cells, we turn to RNA sequencing. So we intranasally infected neonates and adults with both wild type genotypes and IFNAR genotypes with flu or sham controls. We then isolated primary type 2 epithelial cells from the lungs at early time points post-infection, so 1, 2, and 3 days post-infection. And then we sequenced them and analyzed them for different gene transcription. So the first thing we performed was principal component analysis, and this would cluster samples based off of transcriptional similarities. But what you can notice is across the x-axis, with a large 68.4% variance, are two groups. And when we look at our data, the group on the left are neonates and the group on the right are adults. And this just underscores the importance of using an age-appropriate model, because you could not use these adult samples to recapitulate the gene expression in these neonatal samples. But the one comparison we were really interested in was the IFNAR neonates versus the wild type neonates, to understand how these type 2 epithelial cells respond differently in the presence or absence of interferon signaling. And when we performed pairwise comparisons at each time point, we found that the highest number of differentially expressed genes occurs at 2 days post-infection, where 84 genes are upregulated and 24 genes are downregulated in the IFNAR neonates relative to the wild type. And for the sake of time, I'm just going to focus on the 84 upregulated genes. Now, the next thing we wanted to do was perform pathway analysis, and this would give those 84 upregulated genes some biological context. And what we found is that the upregulated genes were associated with tissue development. Now, we expect tissue development to be occurring in our neonatal model because, again, they fall within this rapid 2-week developmental window. However, in our model, when we layer flu infection on top of it, our data suggests that the type 1 interferons produced in the lung in response to flu infection feeds back and blocks lung development in the wild type mice. But in the IFNAR neonates, where we take away this type 1 interferon signaling, lung development can continue on as normal. So we wanted to look then at specific developmental genes and how these changed in response to infection based off of interferon signaling. And what we found was this interesting pattern, where at baseline, these genes are expressed similarly between two groups. But in response to infection, the IFNAR neonates upregulate these developmental genes while the wild type downregulate them. And I just chose two genes here. One deals with vitamin A signaling and one deals with Wnt signaling. Both are associated with lung development, but in the context of flu infection, they can also be associated with lung repair. So we further hypothesized that the IFNAR neonates would be better able to repair their lungs in response to infection. So we looked through our epithelial cell sequencing data to find more evidence of genes that related to lung repair or lung development. And that led us to tight junctions. So tight junctions are physical connections between neighboring cells. When those are disrupted during flu infection, you get increased immune cell infiltrate, increased fluid influx into the lung, and it's associated with severe pathology. And what we found within our type 2 epithelial cell sequencing data is that the IFNAR neonates maintain this tight junction gene Claudin 8 over the course of our time course, while the wild type start to lose Claudin 8 expression by 3 days post-infection. But from our model, we don't really see mortality until 6 days post-infection. So we wanted to look at what happened to Claudin 8 gene expression later in infection, and we also wanted to expand out to the whole lung. So we performed targeted PCR looking at Claudin 8 gene expression. And we compared the gene expression in our infected samples to uninfected controls, and that's shown by this dotted line here. And I really just want to focus on this 6-day post-infection time point, where you see that our IFNAR mice in gray are better able to maintain expression of Claudin 8, while the wild-type neonates have virtually no expression of Claudin 8 in the lung. So we next wanted to ask the question of how does type 1 interferon signaling impact tight junction integrity late during infection? So we see it in our transcriptional data, and we noted Claudin 8. So Claudin 8 is an occludin co-recruiting molecule, and occludin is a very key tight junction molecule for sealing of the alveoli. So we wanted to look at occludin expression, but also expression of the anchoring molecule ZO1 at 6 days post-infection. So we performed immunofluorescence. And what we found is that there was no significant difference in ZO1 expression between the wild type and IFNAR neonates. However, we do see a significant difference in occludin. So you can see that the wild type neonates have very limited occludin staining, while the IFNAR neonates are better able to maintain this expression. And we were able to quantify the reduction in occludin due to infection by comparing the mean fluorescent intensity from an uninfected control to the 6-day post-infection. And we do find that the wild-type neonates lose significantly more occludin, around 60% compared to the IFNAR. So when we take all of this data together, what it suggests is that the type 1 interferons produced in response to flu infection in the wild-type neonates leads to a decreased expression of cell repair genes early in infection, leading to decreased tight junction expression late in infection, and we observe increased lung pathology and decreased survival in these mice. And in our IFNAR neonates, where we are able to block type 1 interferon signaling, we're able to see increased expression of cell repair genes, increased tight junction expression or maintenance of tight junction expression, coupled with reduced lung pathology and overall increased survival. So I just want to thank the Carey Lab and all of the present and past members who contributed to this project. And I also wanted to thank Dr. Josh Mell, who was our bioinformatics collaborator on the type 2 epithelial cell sequencing, as well as the Genomics Core facility at Drexel and the Sidney Kimmel Cancer Center, who performed the actual sequencing of the type 2 epithelial cells. With that, I'll take any questions.
Very nice talk. I have a couple questions. So when you look at I'm sorry if I missed it. If you look at the neonates, the IFNAR knockout versus the wild type, was there a difference between the viral load in those?
There was no difference between viral loads. So despite the fact that we took out type 1 interferon signaling, the neonates still had similar viral loads. So there's some other mechanism for viral control.
Yeah, so it goes to that. Have you, in the RNA-seq data, did you look at the interferon lambda expression as well as maybe the type 1 interferon, how much of a difference in that when you
Yes.
took out type 1?
So that is actually the future direction of this project, is focusing on type 3 interferons. So we actually have a slide. Let's see if I can get to it. So we did also use lambda knockout neonates, and we find that if you take out lambda, the neonates have exacerbated mortality compared to the wild type. And then if you use a double knockout, it's similar to the single knockout of lambda. So we think that type 1 interferons are solely pathogenic, and the redundancy of the lambda and type 1 kind of functions serve to be just solely protective in the neonate. So that is where this project is headed, yes.
And in terms of the expression of the ISGs, comparing the lambda versus type 1?
The knockout, the lambda knockout.
Yeah.
So that is one thing we're looking at. So we do have sequencing studies going on comparing the IFNAR lambda knockout and the wild type to compare all of those things.
Thanks.
Nice talk.
Thank you.
What is the status of C-gas sting signaling in these cells? And what does the mitochondrial physiology look like when you have the infection, whether or not that has any impact on the overall death and that you don't get that damage occurring?
So we haven't looked specifically at the CGAS sting pathway, like activation of the mediators. As in terms of the mitochondria, I do think that the mitochondrial ROS may have something to do with the susceptibility of the IFNAR and possibly leading to cell death.
Or the release of mitochondrial
Yeah.
DNA
Yes.
that may lead to the whole.
So we're
situation.
investigating mitochondria as well in terms of like cell death activation or reactive oxygen species production in these IFNAR mice versus the wild type.
Thanks.
Okay, thank you very much. And I would like to welcome up our last speaker, Kira Walasek?
Willis-Tech, yeah.
Walasek. Sorry, is it this one?
Should be, yeah.
Yeah, there we go.
Beautiful.
All right.
Thank you. All right. Thank you everybody for coming out. I'm really excited to show you guys some of the research I was doing for my PhD, which I defended in April. So this is my first talk as Dr. Kira Walsh. It's very exciting. Perfect. So today I'm going to talk to you guys about early immune events during SARS-CoV-2 infection and how they impact memory T and B cell responses. So memory immunity to SARS-CoV-2 can be established through either infection or vaccination. And after these antigens are present in the system, they are picked up by dendritic cells or other antigen-presenting cells, which will then travel to the lymph node. And in the lymph node site, your antigen-presenting cells will present to CD4 T cells and naive CD8 T cells. Now, when we look at what's actually going on in the lymph node, try to bear with me in this figure. We're not going to go too deep into it. But when you have your dendritic cell, as I mentioned, it'll present to naive T cells. And once this happens, your CD4 T cells have a few different directions it can take. They can become CD4 effector T cells and undergo proliferation, They can also go down the path of becoming CD4 memory T cells. Additionally, they can differentiate into T follicular helper cells, which is what our lab is interested in. And TFH cells have the important role of presenting antigen to naive B cells. And this interaction is very important for establishing better quality and quantity of antigen-specific antibodies, particularly neutralizing antibodies as well. So your naive B cells can then enter into germinal centers where they will undergo affinity maturation. They can also differentiate into memory B cells or long-lasting plasma cells. And from here, they will exit from the lymph node into the bloodstream, and that's where these cells can interact with or produce different neutralizing antibodies upon reinfection with SARS-CoV-2. Now, what is the importance of memory immunity? I think when people talk about SARS-CoV-2, we're really interested in things like your cytokine storm, like when you're hospitalization, or how you're making antibodies. But a lot of papers have not looked at long-term memory immunity past the first few weeks. And memory immunity is important because it establishes rapid and effective clearance of a pathogen after re-exposure. And I think we're all familiar that during the SARS-CoV-2 pandemic, this is incredibly important as we had multiple variants of concern coming out and quite quickly as well. And it also minimizes damage to the host, prevents severe disease, hospitalization, and death. So this led us to our main question of what factors during early SARS-CoV-2 infection contribute to the strength of memory immune responses? So to do this, we were part of a nationwide study called the IMPACT study, and this encompassed 20 different universities across the United States. And just a brief overview of this clinical study, participants were recruited when they were hospitalized with COVID-19 and they were affirmed COVID positive using PCR tests. And this study started at the beginning of the pandemic in around May of 2020. When patients were admitted into the hospital, they had their intake day, and then biological samples were collected from day 0 to day 28, and we refer to these time points as our acute time points during hospitalization. Participants then came back for 4 longitudinal time points, including 3 months, 6 months, 9 months, and 12 months. And I would just like to point out that in between these 2 time points, month 3 and month 6, this was around winter of 2021, and this is when participants started receiving the mRNA vaccines. When participants came in, as I mentioned, different biological samples were collected. Most of them were blood, but during these acute time points, we also had sputum and nasal swabs that were taken for nasal viral loads. And these different samples were then sent to different core facilities across the United States where different tests such as autoantibodies, transcriptomics, and metabolomics and flow cytometry were taken. So one of the first things we also looked at that was part of this study was patient trajectories. And not everybody who went to the hospital with COVID-19 died. I think early in the pandemic, a lot of people, when they started feeling sniffly, they would go to the hospital just to make sure everything was okay, especially people who had comorbidities as well. So patient trajectories were established, and these were established during the acute phase of hospitalization, so during the first 28 days. And the severity of illness was determined using a 7-point ordinal scale. And then from this, they were differentiated into 5 different groups. So we had trajectory 1 to trajectory 5. And the patients we'll be looking at, and on my study today, we only had participants up to trajectory 4. Trajectory 5 patients, unfortunately, were ones who passed away, so we don't have longitudinal samples to look for memory T and B cell responses for them. So one of the first questions we had was, does disease severity impact memory responses? And I would just like to point out what I very previously mentioned is that in our study of 46 participants that we're looking at in this study, this was kind of the breakdown. So most of them were trajectory 1, which is, mild illness. They went to the hospital, they were out in a few days, versus trajectory 4, which is, mechanical ventilation and severe COVID-19. So as I mentioned, one of the first things we looked for was if patient trajectory influenced the strength of memory B and T cell responses. And when we ran correlations between different memory responses, such as antibody responses, memory B cells and memory T cells, we didn't see any correlation between these groups. Also, the data not shown here was we also looked for correlations between gender and age as well, and we didn't see any significant correlations for those groupings as well and their strength on memory immune responses. So next, what we did was we looked at using flow cytometry, we looked at the frequencies of RBD specific memory B cells 1 year after hospitalization. And as you can see here, there's kind of a spread in what we're seeing. You have some patients who are high memory B cell responders, and you have some patients who are low memory B cell responders. So once again, we looked at correlations. So what we did was we looked at correlations at early time points, and at day 0, these patient samples were used for O-link analysis, and we had a 95 panel of different inflammatory cytokines and chemokines that we were looking at. So we correlated all of these different protein O-links with the frequencies of memory B cells, and we saw a few positive and negative correlations for this group. So overall, we're seeing pro-inflammatory cytokines and chemokines such as CCL23 and CXCL10, which are positively correlated with the frequencies of memory B cells. And we also see a negative correlation here with this FLT3 ligand. And this is a ligand that is found on dendritic cells, and it's mainly upregulated for DC maturation and differentiation. So next, we also wanted to look at the frequencies of CD4 positive T effector memory cells. And to do this, we used an AIM assay. And in an AIM assay, we're just looking for upregulation of different surface cell receptors on these CD4 T cells. And once again, similar to what we saw for the memory B cells, you're not seeing a distinct differentiation between these participants. Some are high responders and some are low responders. So once again, we ran correlations with early immune proteins to see if any of them were positively or negatively correlated with memory T cell responses. This is just a table showing all of the positive and negative significant correlations that we had. And I would just like to point out that we're seeing a similar trend with our memory B cells, where we see pro-inflammatory cytokines such as IL-33, CXCL13, and CCL2. So this one is negatively correlated and these two are positively correlated. However, we're also seeing a negative correlation with this FLT3 ligand, which I mentioned is important for D cell maturation. Next, we looked at the circulating IgG neutralizing antibodies to SARS-CoV-2 variants. And in this study, we looked at the variants Wuhan and Alpha. And over time, you can see that there's a sharp increase in the beginning. However, overall, it does remain steady. And once again, we're seeing this trend where participants are either high responders or low responders. So similarly, what we did, we're trying to establish which pro-inflammatory cytokines or chemokines are either positively or negatively associated. And once again, we see this trend of pro-inflammatory cytokines that are positively associated, including interferon gamma, IL-6, IL-18. And we're also seeing important T cell receptors such as PD-L1 and CD40 as well. Additionally, we also wanted to look at neutralizing antibody decay. So it's one thing to be a high responder for neutralizing antibodies at a certain time point. However, we were also interested to see the rate of decay of these neutralizing antibodies. And just briefly, these are all of the participants who had neutralizing antibodies at multiple time points. And we looked at the 3 different strains in this study that we had. And what we're looking for here is a sharp slope, indicates that there was a higher degree of decay, and a flatter slope shows that this participant had a longer lasting neutralizing antibodies. And one thing I'd like to also point out is that the half-life of the Wuhan neutralizing antibodies are correlated with different subsets of monocytes as well as plasmacytoid dendritic cells. Now, different monocytes and different dendritic cells have different roles, especially in viral infection and COVID-19 infection. So we were interested in the CD14 DIM monocytes, which are pro-inflammatory. And we also wanted to look at the different subsets of dendritic cells that were important for viral infections, such as PDCs and MDCs. And both of these are pro-inflammatory and the main producers of type 1 interferons during infection. So when we correlated these as well, we saw positive correlations between myeloid dendritic cells, plasmacytoid dendritic cells, and RBD IgG titers. Additionally, we looked at the transcriptomics that were taken from these early time points between O-link, and we also looked at different immune pathways that were correlated with these memory immune responses as well. And we see that type 1 interferon responses are negatively correlated with T follicular helper cell memory cells. And then we also saw that interferon gamma responses were positively correlated, which, as I mentioned before, is kind of going along with this really nice theme that we're seeing. And these are some really lovely integration plots that are just kind of showing the overall correlations that we found between all of these groups. So these are all of our positive correlations that are shown in this river plot. And as you can see, that we took into account different proteins from both Luminex, O-link, and then from our transcriptomic data. And the darker the color, the stronger the correlation. And some of them are correlated with multiple different types of memory responses, such as Cas8, and our largest one was CXCL10. Additionally, we have negatively correlated proteins as well, not as many as the positive correlations, but once again, I'd like to point out that this FLT3 ligand, it was of most significance and important to us in this study. So this is just kind of an overall schematic of what we think is happening and the different cytokines and chemokines and how they're affecting different memory cells and different memory responses. So as I mentioned, SARS-CoV-2 comes into the cell, And then in response, you have different inflammatory cytokines, ISGs, type 1 interferons, type 2 interferons that are being produced by different cells. And these are the responses on the different memory cells or neutralizing antibodies that we found in this study. So our overall conclusions are that high levels of inflammatory proteins and co-stimulatory molecules during early stages of COVID lead to enhanced memory T and B cell responses. We also saw that inflammatory response pathways are positively correlated with B cell, T cell, and neutralizing antibodies. As I mentioned, type 1 interferon pathways and proteins are negatively correlated with neutralizing antibody responses. And the durability of neutralizing antibody responses is also negatively correlated with type 1 interferon producing cells. And with that, I would like to thank my lab, as well as the Kutzler lab, who has been a great help as long as with our lab, the Haddad lab. And I'd like to thank our collaborators, Slim Farati at Northwestern. He is our bioinformatics guy, and he has been an immense help on this work, as well as the Sanders lab at UMC, who has done all of our neutralizing antibody studies for us. Additionally, I'd like to thank the impact study, Tower Health, and our funding sources. And I'll take any questions.
Kara, great
Okay.
talk. So what lessons would you take from your studies as far as SARS immunization or vaccination
Mhm.
to have better memory, better neutralizing, and also what would you suggest that would be required for that?
Yes, so for better memory responses, especially in mRNA vaccination, we've kind of discussed future directions of including adjuvants. So based on this data that we see here, CXCL10 is a very strong sting inducer. So we actually collaborate with one of the people on my committee, actually. He's kind of like a sting vaccine biochemist, and using CXCL10 as an adjuvant has kind of started to be looked at in vaccinations to help stimulate different pathways that the mRNA wouldn't be in the cell. And hopefully that would be a really nice future direction for looking at stronger memory T cell responses. And also based on this data, because it was our highest correlation and with many different memory cell types.
So do you think using antigens rather than mRNA in combination with some
Yes,
other
I think adjuvants in combination would be an interesting study to look at with the mRNA vaccine, because we know that they're safe and they've been very well established, especially as we're still adding, different strains of COVID-19 yearly into the vaccines.
Did you look at all at which vaccine the patients received and if there were any differences between the manufacturers?
Yes, so the participants that were included in this study, and also like the other part of my thesis, we only looked at patients that had mRNA vaccines. So we excluded any patients who had like the J&J, or we didn't have anybody with Novavax, but there was no protein subunit vaccines. However, we did not tease out differences between the Moderna or Pfizer. Honestly, most of the immune responses are pretty similar, and that's been pretty well established between the two vaccinations as well. Yeah.
Thank you so much.
Cool. Thank you.
Okay, before our next talk, oh, sorry, I don't know why I can't unshare. Okay. Oh, you're already sharing. Oh, perfect. Okay, sorry. Before our next talk, which will be the Toby Eisenstein Memorial Lecture given by Dr. Becky Limison, the president of EPAASM will say some words about Toby. Thank you.
Thank you. So first, I want to congratulate the student chapters and its advisor, Sunny, for organizing another successful May meeting. And thank you for the trainees for giving terrific talks. So I can't think of EPAASM without thinking about Toby Eisenstein because she was the cornerstone of this branch for so many years. Toby was a Philadelphia native. She went to Philadelphia High School for Girls before going to Wellesley College for her bachelor's degree. She received her PhD in microbiology and immunology from Bryn Mawr, and she married her high school sweetheart, Bruce, for 60 plus years. And we're honored to have Bruce here tonight to join us. Toby joined Temple University as an instructor in 1969. She rose through the rank, became the professor in 1984 in microbiology and immunology. Her research focused on cellular immunity in salmonella infections, mechanisms of Legionella infections, as well as contributed to efforts toward developing Group B strep vaccines. In 1989, she collaborated with Martin Adler and to look at the effects of morphine and other opioids on the immune system, which resulted in the establishment of the Center for Substance Abuse Research. She also contributed a lot to both National ASM as well as a branch. She was a member for 57 years. She was the president of this branch from 83 to 85, and she remained very active within the branch in various activities. She was the fellow of the American Academy of Microbiology, as well as the chair for the branch organization committee that really helped institute a lot of policies that support the branch. She was also the chair of the membership board in 2003 to 2012. Again, she helped support the branch as well as supporting students and postdocs. She also supported the ASM archive program. She appointed the late James Poupard to help with the ASM archives. She also oversaw different activities for minority committee as well as the training and career activities. She was the founder of our student branch in 1996. Back then, most branches student chapter from one institution. She proposed that we draw different students populations from the local university and medical schools. And so our chapter was unique in that it was multi-institutional and mostly made up of the graduate students. So on a personal note, Toby was a brilliant scientist. She was a kind and generous colleague, as well as an exceptional educator. I had the pleasure of co-directing a course with her, as well as co-taught a lot of medical school workshops. I always look forward to those sessions, not just to teach, but also to catch up with Toby about our lives, about our families, about science. We bonded over the love of microbiology as well as love of classical music. She was a enthusiastic patron of Academy of Vocal Arts as well as other classical music. I started playing violin once I was 6, and my wife is an alum of Wellesley. Anyone who knew Toby also knew that she had the best stories of various major discovery in microbiology. It helped that she was involved in a lot of those seminal studies. She also didn't shy away from sharing her experience as a woman in science. The struggles that she had to face balancing a demanding career in academia as well as raising a family. She shared those stories not just as the reflections of the challenges she and other trailblazers faced, but also as a reminder that this fight for equal opportunity continues. And so, as I mentioned, she founded this our student chapter, and we will celebrate her legacy next year when they have their 30th anniversary. So I cannot think of anyone more deserving for this honor. And it is with deep respect and admiration that we dedicate this lecture as the Toby K. Eisenstein Memorial Lecture. So now I would like to turn over to the students to introduce our speakers. Thank you.
Okay, Vincent, thank you so much for the kind words about Toby. Our keynote speaker tonight will be Rebecca Lamison. She is a faculty member at MIT, where she studies, or where her lab studies host virus interactions, specifically on cell-to-cell spread of very common and very important pathogens such as Rickettsia and Listeria monocytogenes. So without further ado, Becky, please come on up.
Do we have a power cord? Because
Yeah, there we go.
fantastic. Make sure that. Okay, we will not be able to see, I think, the mouse, but it should be fine. All right. First of all, thank you so much for this invitation to give this talk. It's been really great hearing all the presentations already. It's been a lot of really fantastic science, and it's a hard act to follow. So I will do my best. And today I'm going to talk about some work looking at how microbes, specifically Rickettsia, manipulates our host membranes. And so we're going to talk actually about a couple of different stories. And I decided to throw in a third story that kind of flips this title too, to ask how a host can manipulate the pathogen membrane. And this is an emerging story as well. So I'll kind of give you an overview of all of those things. But first, let me just say that some of the questions are really interested in the lab is, we have all this diversity of microbes that we interact with, and we really want to understand kind of all those different mechanisms that are used by pathogens to interact with our cells. And as Dan mentioned, we are interested in a couple of different types of bacteria, including Listeria and Shigella, which we get from food, and also Rickettsia, which we see from these tick-borne pathogens. And why we pick all of these random bacteria to kind of study, they actually all share a very similar life cycle in that they're cytosolic bacterial pathogens. And so they have access to the same space, and yet learning how they differentially interact with that space, I think, is incredibly fascinating. And the reasons we do this is because we, of course, want to understand their molecular pathogenesis. Why do they make us sick? And is that useful information that we can use therapeutically? But also, these are tiny cell biologists. And so we can learn a lot about how our host cells function by looking at how this function might be dysregulated in the face of a pathogen. So for today, we're going to focus on primarily what most of my lab actually studies, which is this pathogen Rickettsia parkeri. And Rickettsia parkeri is part of the spotted fever group of Rickettsia, which we see in the United States. This group actually has the deadliest tick-borne disease known to man, Rickettsia rickettsii. We don't study that one. We study parkeri because we want to be able to work with it safely and easily in the lab. And parkeri is a really nice model for this more deadly variant. Importantly, both of these organisms, or many species in this genus, are actually on the rise in the United States. And a lot of this has to do with the fact that the ticks are migrating northward. So you can see where you are now. It's clearly covered. And I think recently Connecticut was hit in October, and my lab keeps a tracker going for when it's going to hit Massachusetts. And it hasn't yet. So we'll see. But it is clearly increasing, and it is something that is very much an emerging and largely neglected pathogen. And one of the things that really sets Rickettsia apart from the other two organisms we study in the lab, like Shigella and Listeria, is that much like a virus, this pathogen can only grow inside vector and host cells. And this presents some really significant challenges. It's really hard to, we can't grow them outside on plates or in liquid culture. And so the tools that are available and the kinds of advances in our understanding are about decades behind a lot of well-studied pathogens. But undeterred, we're really fascinated by these pathogens because this exclusivity has created these really unusual adaptations that we keep discovering by looking at how they're interacting with our cells. And that's something that I really want to highlight for you today. And our overall goal is to understand kind of at a systems level and at a cellular level, what are the host and bacterial factors that are driving these kinds of interactions. So Rickettsia parkeri is one of these, as I said, cytosolic bacterial pathogens, and it has a pretty dynamic and complex life cycle. You can see that it can get into cells and grow within this cytosol. It actually enters pretty rapidly into any type of cell we've thrown at it so far. Once it gets in that cell, like I said, it's going to grow and replicate. And then one of the things it can do is it can hijack actin. You can see that in green. It makes these actin tails that actually helps them rocket around inside the cell. And Rickettsia are the only bacterial species we know so far that can make two different kinds of tails at two different times of their life cycle. That's weird, right? It's an obligate pathogen which has stripped down this genome, and yet it's maintained these like two very distinct types of motility. And that's not the only times that we sort of see these unusual adaptations. And so when I started as a postdoc, I had never heard of this pathogen, and I very naively thought this was a very interesting system to work into, not knowing some of the challenges. But I was just super fascinated by how they were kind of interacting with our cells. And one of the things that really grabbed my attention is this spot here in the middle where the pathogen is actually moving from one cell to the other. It's actually manipulating this cell-cell junction or this plasma membrane. And we've done some work to kind of understand this process, and this highlights one of the big questions we've been thinking about, just globally. How do these pathogens manipulate our own membranes to facilitate their infectious life cycle? And I'm not going to go into this data here, but we've published a couple of stories, highlighting some of the ways this has happened that I think highlights the ways that we think about many of these processes. So for example, we've identified that there's a secreted effector called SCA4 that can be delivered into the donor side of the cell, and it can change the properties of the cell-cell junction to make it easier for this pathogen to remodel this cell-cell membrane or cell-cell junction and create this protrusion. And then they can also, we recently learned, can recruit clathrin to this site to help them be endocytosed into this space. And that's largely, as a field, what we've thought about is how do they manipulate the plasma membrane, either when they're getting in or when they're trying to spread. But if you all know, your cells are not gray boxes, right? Like they're chock full of lots of stuff. And I think as a cytosolic pathogen field, we often forget to think about the contents within our cells and what are the things that they're potentially interacting with. And there's a lot of options here. And in fact, of the membrane compartmentalized options, the largest one is the endoplasmic reticulum. And so I want to tell you a story a little bit that we've done trying to understand this potential for an interaction between this cytosolic pathogen and the ER. And this is something we just recently published, so I'll just kind of briefly go through some of that. And it'll also let us then think about how do they also interact with our nuclei, which is something special that these pathogens can do. And then once we talk about sort of these host membrane interactions, I'll flip the script and talk a little bit about how we're looking at the bacterial membrane as well. Okay, so to get started with this ER story, we did what any good cell biologists do, is we went to the microscope and looked at our infected cells. And so what Yami did here, as she was the lead author on this story, is she labeled her host cells with an ER marker. So you can see this really beautiful ER network in cyan, and then you hopefully can see a few magenta bacteria in this space. And you'll notice the one that's boxed here, there's an example where you can see a little bit of that cyan membrane wrapping around the around the rickettsia. And that was kind of weird because these are cytosolic pathogens and they really shouldn't be wrapped by the ER. And yet we were seeing maybe 5% of the population was wrapped by this membrane. We also noted that the ones that were interacting with the ER weren't really moving around. And so we know that with wild types bacteria, a lot of them can make these actin tails and they could move around, but not all of them that were engaging in this activity were moving. And so we wondered if actin-based motility was somehow negatively regulating this process. So if we could infect with a strain that can't move, would we see an increase in these interactions? And fortunately, we do actually have a mutant. We'll go back to that. Here we go. That cannot make that actin tail called a SCA2 mutant. This SCA2 protein will come up later. And you can see all of a sudden now we have a huge increase in these interactions because these pathogens can't move around. And so we wanted to get a better sense of what is the nature of this interaction, because the confocal is lovely and it's showing us this really interesting interaction with the membrane, but we can't really tell like how much is wrapping, how well connected it is to the network, and are they in the ER or are they actually being wrapped by the ER? And so for that, we did electron microscopy, which you can sort of see here, where we've marked the rickettsia with an R and the mitochondria with an M. And if we zoom in and color code the things that you need to worry about, you can see very clearly that the bacteria in magenta are not inside the ER. They are in fact being wrapped by the ER membranes. And you'll notice that a good perimeter of the bacterium is actually being covered, and that's pretty close. And also what you can't maybe see so clearly with color coding here, this is the rough ER. It's very specifically that interaction. And so this gave us some first initial clues that the bacterium were being sort of wrapped by this ER network, but what we can't see in this picture is how much of the rest of the network is part of this interaction. And so we decided to use something called FibSEM, which is essentially just making a three-dimensional block of an electron microscopy image where you're taking this image and then you sort of laser off the top layer, and then you take another EM image, and then you do that serially, and then use a computer to basically rebuild that. And then we use deep learning segmentation to identify the components within this block, so specifically the ER and rickettsia. And if we peel away this block and reveal, oh, it's very jerky. Okay, it's happening now. We peel away this block, you can see that the ER membrane is presented, and as we sort of peel that ER membrane away, I want you to notice two things. One is that the bacterium really are getting wrapped pretty significantly by this network. It's not completely, they still have access to the cytosol, which is kind of interesting. And these interactions are still connected to the greater ER network too, which means they potentially still have access to this larger space. And so why am I showing you all this? Well, it turns out that our ER has this, I think especially in the last couple of decades, has been appreciated for not being these isolated entities that our cell biology textbooks often would show us. In fact, it makes a lot of interactions with our organelles. And one of the ones I'm showing you here has garnered a lot of attention, which is the interactions between the ER and the mitochondria. And this is kind of notable because rickettsia and mitochondria have had a long storied history, potentially evolutionarily linked. And so what do we know about these kind of connections that are interesting? We know that these, what are called membrane contact sites, have been very well characterized between the smooth ER and mitochondria. They have a very defined distance between 10 and maybe 30 nanometers, so they're very close, and they have very specific tethers. There's a subset of these then that are also found specifically with the rough ER. They tend to have a longer distance closer to that 80 nanometer region, and they look a lot like our interaction, right? You can see this rough ER membrane kind of wrapping around this mitochondria. And those specific rough ER interactions have been characterized in so far 3 papers. So there's not a lot that's known broadly. They're called a variety of things like Merck's for mitochondria ER contacts. And so we started to wonder, were our bacteria mimicking Merck's? And so we asked if these were Merck's, if these were bacteria ER contacts. And so we tried to use the same parameters that were used to define these kinds of mitochondrial contacts. This included measuring the distance between these spaces. And so Yabi very carefully went in and measured the intermembrane distance in her EM images. And she found that there was a very similar distance between the rickettsia and the mitochondria here, suggesting that they are held at the same distance. If they're held at the same distance, then obviously they might be using similar tethers. And so one of the ones that she became particularly interested were these tethers shown here in orange that are found on the ER membrane called VAPs. And there's 2 of them, VAP-A and VAP-B, that are really important ER tethers for a lot of these contacts. And those VAP proteins have been found to be hijacked by other pathogens, but specifically pathogens that live within a host-derived vacuole compartment. An example here is chlamydia, where it's using this VAP to sort of bring ER close to its vacuole. And so we did a variety of experiments to show that VAPs were localized. I'm just going to show you one set of those experiments. This is looking at the endogenous protein. So she's using immunofluorescence here. And actually, I think maybe Patrick did this experiment looking at VAP-A. And you can see very clearly in the top and in the bottom rows, VAP-A is localized around the bacterium, where we're seeing co-localization with the ER as well. And we're seeing this not just with VAP-A, but we can also see this with VAP-B as well. And not only is it localized, it turns out we think it's essential for this interaction. So we were able to get our hands on some VAP-A, VAP-B double knockout cells from Pietro De Camilli's lab, and we were able to show that this completely abolished this interaction. So this seems to be the critical tethers for driving this space. So we still don't know why these are happening, and I'm sure this is something you all want to know too. We understand some aspects of this interaction. It's, first of all, the first example, as far as we can tell, of a direct interaction between the rough ER and a bacterial surface, sort of making one of these bacterial mimicking, or bacteria mimicking one of these like membrane contact sites in the cell biology field. It seems to be negatively impacted by their motility. So if the pathogen can't move, we see more of these. We've been able to identify on the host side the factors that are critical for this process. And what we know so far is that it's not obviously correlated with fitness. You know, if the bacteria are making this interaction or not, the bacterial burdens are identical, at least under the cell types and conditions we've tested so far. We have not been exhaustive, so we are still very much interested in this question. We've also shown that it has nothing to do with autophagy. So this is not a way for the host to try to clear this pathogen. And so we have a lot of work cut out for us, right? We have this novel interaction that has never been described before, and we have a lot of questions about how it's happening and why it's happening. And so one of the things that Caroline is continuing to look at is, you know, now that we know the ER tethers, can we identify the targets of those tethers on the bacterial surface? And this is an important question because we actually don't know all the constituents of the bacterial surface either. And so we can imagine that VAPs could be targeting something either in this putative S layer shown on those green blocks at the top, or targeting some other bacterial proteins directly. We could also imagine this is a host protein that's mediating this interaction too on the bacterial surface. So she's got some mass spec data, I think, coming in the next day or two to let us know if she's actually found this target. And I think that's going to be one of the keys to helping us unlocking the functional consequence of this. Another way that folks often define these functional contact sites is defining what are the actual unique proteomes that are found at these sites, because these contacts are not just a random connection. These actually facilitate very specific functions that are uniquely defined by these contacts. And we can figure this out a lot of times by figuring out what are the proteins that are enriched in this site. And that's something we're also trying to work on as well. And what this has been, you know, really nice to be able to think about is that, you know, our bacteria are not just hanging out in isolation in our cells, and there's a subpopulation that is able to start to make these connections with some of these organelles like the ER. And this story also started to bleed into this other story we were thinking about too, which is what other compartments and places can these bacteria go? And this is a story that Hannah has been working on, and this is currently unpublished, looking at how these bacteria can actually get inside your nuclei, right? And this has been a really fascinating story. It's often something that when people work in my lab, they'll go to the microscope and they'll note that the bacteria are in their nuclei, and they're very confused by this. And nobody has been brave enough to kind of tackle this. But Hannah was extremely intrigued by this process. And we're not the only ones that have observed this invasion. It's actually a century-old mystery. So back in 1919, Wolbach first described this. You can see a drawing here on the left of a tick epithelial cell with this enlarged nuclei with a bunch of bacteria inside of it. And then we've seen this across many other different types of mammalian and tick cells. And it was for a very long time too a diagnostic. You could use this to say if you had a spotted fever group infection versus a typhus group infection. So we knew it existed, but we didn't actually understand how it was happening. And if you read a lot of the literature, a lot of folks just assumed model 1 was correct. This model that the pathogen is already moving around, and maybe during mitosis, the nuclear envelope is breaking down, and the bacterium are just getting trapped when it kind of reforms as the cell cycle progresses. Alternatively, model 2 is a possibility as well, where the bacterium are actively pushing against this double membrane structure and invading the nuclei. You can probably guess which one I favor. And it's really going to be model 2. And we have, you know, a lot of evidence to support this. So first, we sort of got these initial images where we've taken, in this case, Hannah was doing fixed cell imaging to define the nuclear boundary. So she's used nuclear lamina to label the nucleus. And hopefully you can see very clearly 2 examples of a bacterium like ramming into this double membrane structure. And behind it is this actin tail that's probably helping it push against this. And so she quantified the frequency. In this case, in this particular cell, this is A549 lung epithelial cells, which is just one of our really nice bread and butter cell culture models for this pathogen. And it happens a lot. It's 26% of the time, which is not infrequent. There are some very, very rare examples of maybe some other bacteria potentially doing this, but it's 1 in 1,000, 1 in a 2,000 kind of phenotype. This is pretty common. So she asked, if you can see some of these arrows pointing to some of these infected nuclei, she asked, is it in fact dependent on actin-based motility? And I've told you we have a mutant that can't move. And so she took that mutant and infected those cells, and none of them made it inside the nuclei. And so I find this a really fascinating kind of pause point here, because we have two very different double membrane structures that this pathogen is going to try to navigate throughout its life cycle. I very briefly mentioned this process of cell-to-cell spread, and one thing I didn't mention was that when rickettsia spreads from cell to cell, it actually loses its tail, and it makes a really short protrusion. And yet when they're diving into this nucleus, they're holding onto their tail, and you can see this kind of long protrusion that's being made. And I find that really fascinating. But maybe that has something to do with the way that it's moving into this space, that something is, maybe it's harder to get into this nucleus and they need a little bit more force to help them in. And so we wanted to look more carefully at, not just that snapshot in time, that single protrusion, but really what's happening over the course of an invasion event to help us understand why this process might be so distinct. And so Hannah did a lot of imaging for lots of weekends to try to capture this process, because it is a lot slower than spread. Spread can be 40 minutes, maybe an hour tops. This thing takes hours long. And so what I'm showing you here is she's caught an event kind of partway through where that protrusion has formed, but now a bunch of bacteria have kind of like come in as well, and they're replicating and they're kind of filling this space. And this is a confocal slice of a movie I'm going to show you in a second. And sort of imagine that ball in the middle is like a balloon that's tethered by a protrusion membrane that's extending from the nuclear envelope kind of far away in space that you can't see. So it's very much that protrusion is formed and expanded. And what she finds is that over time, they're just like pushing and pushing and pushing against this balloon and really trying very hard to break out of this space. And then eventually they do break out. You'll see some of those pink, hopefully guys, near where my arrow is. Once they're inside the nucleus, they then can hijack actin in that space, kind of showing you that picture from earlier. And this allows them to just spin around inside your nuclei, which is the wildest thing that we've ever seen, obviously. But they don't hang out there forever. They do eventually leave. And so you can see some of those events starting over here on the, oh, my arrow is back. You can see the snapshot where she's labeled the nuclear lamina, and we've got a group of bacteria, and they do tend to leave in groups. Again, it's kind of a strange phenomena, but they're coming out, they're using actin tails to kind of push against this space. And if we play this movie here on the right, you can see a bunch of those groups are trying to make their way out, and they're really extending dynamically these long protrusions, really away from this nuclear membrane. And you'll see in the upper right corner, one of them will snap and release the bacterium into the cytosol, right? So that's wild. And it's wild because, too, the cell seems fine. It doesn't seem like this is a major problem for it. It's not immediately dying. And so there must be something about this process that is both mediated by changes in the molecular mechanisms of, like, say, the host or the pathogen. And we're very interested in looking at the host side and the pathogen side. And she's done some work to define some features of the host. For example, there doesn't seem to be a lot of lamins associated with these protrusions. Those seem to get lost when they're invading the cell. The membrane also seems to be devoid of nuclear pore complexes, which is also kind of a strange phenomena. But to get onto the pathogen side, we've been thinking about this process, about what kinds of pathogenic factors might be promoting this particular invasive process. Because again, it's something we see a lot more frequently with the rickettsia, so maybe it's using a rickettsia-specific factor to get in, because lots of other pathogens move around inside your cells, like Listeria and Shigella, and yet we don't see them diving into your nuclei. And so one of the things we've been very curious about are the role of secreted effectors in this process and really broadly at any process inside the cell. And I think this is a really fascinating question. It's also a really challenging question because the identity of the secretome is actually not well defined. And so this has been a challenge in general in the field. And so about the time that we were kind of working on this, we were trying to solve this problem of how do we find these effectors so that we can then start to connect them to some of these cellular processes that we've been studying. And so I want to just tell you kind of how we solved this problem. This was work that was done by Alan Sanderland and also with Hannah's help to essentially, during infection, identify secreted effectors. And we needed to do this because a lot of these are probably type 1 or type 4 secretion system effectors, which you don't really need to know the details. Essentially, it's not easy to predict them. You can do this for other kinds of systems, but for these systems, they don't have highly conserved signal sequences. And a lot of these don't have eukaryotic-like domains that you can use to sort of rank them as potential candidates. And so Alan was particularly motivated to solve this problem. And so he used this technique called BondCat. And essentially what he's doing is that, as the bacterium are secreting proteins into the host cell, they're getting vastly outnumbered by the host proteome. And we've tried to mass spec that the whole proteome and see if we can enrich or identify bacterial factors, and they're definitely outnumbered. And so what he did is he engineered rickettsia to express a mutant tRNA synthetase on the left. And this allows you to add a non-canonical amino acid, in this case, an analog of methionine, so that only the bacterium, and really because it's in an infected cell, only the bacteria are going to be able to incorporate this analog into their proteome, so that when it gets secreted into the host cell, we can then use click chemistry to label bacterial proteins that are in the cytoplasm. And this, what's really nice about this is we can use that click chemistry to add things like biotin to help us fish them out, or fluorophores if we want to see them. And so I'm going to jump way past all of that work that he did and just show you some of the results. We were able to find known secreted effectors, which was really fantastic. But more importantly, we wanted to find new things. And he did find 7 new things. Let's see if this will load. Here we go. 7 new secreted effectors that we're calling SERFs. And with the exception of this first one, SERF-A, that seems to be really homologous to amidases, the rest of them lack structural and sequence homology outside of the Rickettsia genus. So these are very much unique bacterial factors. And you can see they have really telling domains like coil-coil domains or transmembrane domains, or even these lovely domains of unknown function. So they don't really tell us a lot. But why am I telling you about this? We got lucky. So we found out that this guy right here, I'm going to tell you about, seems to have a role in this nuclear invasion story, which I think is really fascinating. And again, this guy has a transmembrane domain and a domain of unknown function 5460. All right, so the first thing Hannah did to kind of make this connection was she just looked at the localization of all these effectors only in infected cells where the bacteria were inside the nuclei. And she found, as you can see, this really striking localization of the endogenous secreted SERF-E protein at these protrusions as the bacterium are trying to make their way into the nucleus. And in another stroke of luck, one of our collaborators actually hit SERF-E in a transposon screen, and were kind enough to actually share this with us so we could start to ask if this is important for this process. And so she was able to show here on the left that the SERF-E mutant, no SERF-E, which is great. And when she looked at nuclear invasion, she saw that there was a significant reduction in how well it was able to get in. We suspect that there's probably more than one factor, and there actually is another one on that list that we know goes to the nuclear envelope, but we don't have a mutant in that one yet, so I don't have anything to connect. But these guys that are even getting in, I want to tell you that they're not happy, right? There's actually an additional layer here that we have to consider. And so one thing that she noted when she was doing some of her imaging was that in this wild type image, you can see 2 time points. On the left, it's 1 hour and 36 minutes into a movie, and then we have 11 hours later, and you can see on the bottom, a cell has had a protrusion form, and the cell at 11 hours later is fine. However, if you look in the SERF-E mutant, you can see that they can initiate this protrusion also, but that cell does not survive. The host cell seems to be needing this SERF-E around to sort of help it maintain its integrity. And in fact, if we look at caspase activation, we can see that the SERF-E mutant has a significant increase in host cell death, and this actually correlates with larger plaques compared to the wild type. And we know that this phenotype is dependent on SERF-E because she can complement it by adding in a copy of the full-length SERF-E here. So this is a really fascinating, I think, way in which this pathogen is manipulating this double membrane structure, which seems to be very distinct for how we've been thinking about the plasma membrane. We are making these really long protrusions with the help of actin polymerization. And we've so far started to pinpoint a secreted effector that we think is acting at this membrane. And she has some evidence that it might even be going to the internuclear membrane. And so far, we think that it might be blocking cell death that would normally be triggered by this pathogen as it's trying to break through this membrane. And our cells have a lot of pathways to sort of sense and maintain the integrity of their nuclei. And so it would make sense for there to be a factor that kind of quells that response so they can successfully make their way in. And we'd also be curious to know, does this also help with their exit? At this point, we don't quite know yet. All right, so hopefully you can see in this first part, we have a variety of different ways in which these bacteria can interact with our membranes. We're thinking about, of course, initially thinking like invasion and cell-to-cell spread. That's attacking the plasma membrane. But we also have now evidence for nuclear invasion as well as Burke's. And each of these represent both dynamic and stable kinds of interactions. And for each of these, we've been really kind of honing in on this idea that maybe something from the bacterium, like an effector, is targeting some kind of host machinery to fulfill this process. And we could also start to think about, too, if there's any surface proteins, but we have yet to identify those just yet. And I think this is still a very fruitful way that we're going to be thinking about these problems. But it kind of misses out on this other side of this equation, which was this third story I wanted to tell you about that's been kind of emerging from the lab. And that is, can the host also regulate the surface? And you might all be thinking of like, of course, Becky, your host cells attack these bacteria all the time. We have antimicrobial peptides or other ways to kind of target and clear these bacteria. But maybe there's positive interactions as well that can help facilitate a rickettsial life cycle. And this is not something we actually knew in advance. And this actually came out of a really fun screen that was initiated by a really talented postdoc in my lab, Brandon Sitt, who was just largely interested in trying to figure out the host factors that are important for rickettsia infection. And so this had never been done before. And so we had to do a lot of setup that I'm going to completely blow past. But essentially, he did a FACS-based screen where he infected a whole genome knockout library in A549 cells with fluorescent rickettsia, and then just looked for cells that had high or low burdens and tried to identify things that might be novel regulators of this process or novel regulators of infection. And he found actually quite a few novel regulators. And when he took some of these and he started to do some initial characterization to confirm that they actually made sense, we found some really nice examples of things we should have expected to see. So for example, the ARP23 complex is important for invasion. And if you knock that out, you can see that the infected cells are much less able to sort of support bacterial burden. So their bacteria are much less able to get into these knockout cells. So that was proof that we were actually finding the kinds of things we would expect. But one of the things that were really striking to us is this hit over here on the far left called PPIA in red, which is our second strongest hit on that side of the graph. And so I want to tell you a little bit about that guy. All right, so PPIA is peptidyl prolyl isomerase A, also known as cyclophilin A. Its job is to help with proteins sort of adopt different conformational changes by creating this cis to trans proline change. And it's also notable for some folks that might think about this. This is the target of the drug cyclosporin A, which is an immunosuppressant. It is also a well-described factor for regulating HIV infection. It actually binds directly to the capsid. And some levels of PPIA are stabilizing and help promote it. And I think increased levels are destabilizing. But the role of PPIA is actually not well understood for bacterial infections and certainly not for rickettsial infections. And so we were very curious to know why we hit this hit. And so Brandon went to the microscope, because that's what we do a lot in our lab, and he looked at infection. And so on the far left, you're seeing the non-target cells. So this is our control infection. And you can see that, as expected, the bacterium have gotten into the cell, and they make those actin tails, so they've disseminated through the tissue. But on the far right, in the PPIA knockout cell, the bacterium seem like they've gotten in, but they're stuck. They're not able to leave that first cell. And this is a really striking phenotype in our field. In fact, it looks a lot like if you knock out the SCA2 gene, the one that I've been talking about this whole time, because it too gets stuck in that first cell. They both can invade, but they can't seem to move away, from cell to cell. And so that made us wonder if PPIA was in fact somehow regulating this process, this ability to move from cell to cell or to move it at all. And so he first then looked at, can our bacteria actually make actin tails in these infected cells or in these PPIA knockout cells? And the answer was no. You can see on the left, we've got normal tail development. Those are those long red actin structures. But when we look on the right in the PPIA knockout cells, no actin tails. So it seemed like something was wrong there. And we had no reason to think that PPIA was regulating actin itself or actin polymerization. We started to wonder if it was actually targeting SCA2 specifically. So let me tell you a little bit about SCA2 and why we thought that. So SCA2, as I've shown you multiple times, is responsible for making these actin tails. It's a special type of bacterial protein. It's an autotransporter, which means it's inserted into the outer membrane and then passes out its functional business end to the extracellular space. In this case, its business end is an actin nucleation region. And so he did what any good cell or any good biologist does these days. He looked at AlphaFold to see if he could see if PPIA actually directly interacted with SCA2. And what he found was that it predicted a very specific interaction with this extracellular C-terminal repeat region. So nothing to do with the actin polymerization zone. It's specifically targeting the CRD region. And if we look on the right, there's actually a loop of the CRD region that's inserted into this pocket of PPIA. And if we zoom in a little closer, we can see that it's predicting a direct interaction with these proline glycine 3 residues, with the residues shown here in cyan for PPIA. And these are notable because this is essentially the same thing that was shown already for HIV that was actually in a crystal structure. And again, you're seeing these proline glycine residues that are highly conserved, targeting this very essential arginine 55. And so we wondered whether or not this protein PPIA was localized to the bacterial pole and actually directly interacting with SCA2, which would be what AlphaFold is kind of predicting here. And so Brandon went and looked at the localization of these proteins. So he first did DS-RED on the top row. So this is sort of our control. You can see it's just everywhere. But when he looked at PPIA fused to DS-RED, you can see a lot of these little puncta. And if you kind of zoom in on the far right, hopefully you can see a lot of those puncta are at the pole of the bacterium that are associated with the actin tail. So it looks like it is actually getting recruited to this site. But then to show that these things are actually directly interacting, he purified PPIA and SCA2. And so in this experiment, I'm showing you a GST pull down with either GST alone or GST fused to just the CRD, that predicted domain that it was targeting. And you can see that when we pull down with the CRD containing GST fusion, we very nicely can pull down PPIA in this pull down. And then he tested whether or not this arginine 55, which has been shown before to be essential for binding capsid, is also important for our interaction. And so we introduced that, and he completely loses this interaction. So it really does seem like PPIA is directly targeting SCA2. But you might be wondering, why is that impacting actin-based motility? So, I did not know a lot about how autotransporters are forming, and this is something that Brandon and I are learning along the way. But canonically, autotransporters go through this multi-step process where this beta barrel shown here in yellow inserts in the outer membrane. And I think some of the models suggest that then part of that protein starts to loop out. And we have here specifically the CRD domain that comes out potentially first. And that there needs to be some kind of a folding event that then drives the rest of the protein out of this beta barrel so it can be presented on the surface. And for a lot of autotransporters, that little loop has a very specific beta strand structure that has an auto chaperone kind of folding sequence. SCA2 does not have that. It has a different structure. And so we're kind of wondering if that maybe this interaction with PPIA sort of overcomes that difference and allows it to help present this protein on the surface, and that there's some interesting dependency that this pathogen has evolved. And so if that were true, then we would expect that not just tails would be gone, but surface exposure of SCA2 would be gone in these PPIA knockout cells. And luckily, we have an antibody that binds to just the passenger region, so we can actually test this hypothesis. And so what he did is he infected his cells, got a non-targets on the top, our PPA knockout cells on the bottom, and we're looking at SCA0, which is a different autotransporter, which does not have this SCA2-like CRD domain. It has its own auto chaperone, and it seems fine. It's on the surface. But in the PPIA knockout cells, SCA2 clearly is not present on the surface. And we can go in and further take these same strains and actually treat them with lysozyme and Triton X100 and show it's not that they don't have SCA2, it's just that the SCA2 passenger domain seems to be inside the periplasm potentially. And what's also interesting, as I mentioned, this is also a target of cyclosporin A. So we see this not just in the knockout conditions, we can also induce this by using a PPIA inhibitor here too. You can see very clearly the loss of actin tails as well as the loss of surface SCA2 on these bacteria. And so this really represents an unusual adaptation for this pathogen to evolve this dependency on an isomerase to help it expose a really important virulence factor. And we're in the process of trying to understand a little bit more about how this is happening. We're also very curious to know, are there other targets of this particular isomerase on the pathogen surface? And also, why? this is a strange adaptation, and I'm happy to sort of speculate on what I think might be happening here. But it just, again, highlights sort of these unusual ways in which this pathogen is co-opting different machinery to support its life cycle. And of course, we're sort of seeing this, broadly across these different stories, that we're seeing this kind of bi-directional manipulation that's happening, where either the pathogen is manipulating the host membrane and the factors on that membrane using a variety of different effectors that we've found so far. And this has been really fruitful for thinking about both the molecular pathogenesis, as I said before, but also sort of how is the host cell functioning kind of in this context. And what this last story helps us do is kind of layer in this different direction. How does the host, again, kind of regulate the bacterial surface? And this allows us to add a third kind of a component of this way we think about this system by thinking about just, also the bacterial cell biology. And are these opportunities too for us to think about how we could potentially engineer surface display if we wanted to in different contexts in a host-dependent way. So these are all things we've been thinking about with these discoveries. So with that, I want to, of course, thank the members of my lab, many of who I've highlighted so far. This work could not be done without an amazing set of collaborators and folks who've just been generous with their reagents and their time. Obviously, funding is also helpful to keep the lights on. But most importantly, I want to thank all of you for your attention, and I am happy to take questions.
Thank you for a beautiful talk. I have a couple questions. So one, as you mentioned, that rickettsia has to do with the plasma membrane when it comes in the rough ER membrane and maybe the nuclear membrane. Do you think it's different mechanism to disassociate from those different membranes?
Dissociate? In
So
what, do you mean when they're breaking out of them?
Yeah.
Oh, yeah. We know very little about what they need to get out of membrane compartments. So there's been a story that came out like a year or two about a factor patatin-1. We have not tested that mutant to see if it's also necessary in these processes. That's a good idea. Listeria, for example, uses two different kinds of phospholipases
Yep.
to help them sort of deal with the differential phospholipid types that are at different membranes. So I would be surprised if that same solution doesn't exist. We just don't have, I think, a lot of mutants in the lab to access that just yet, but that would be good to kind of ask that question, yeah.
And the second question is, in terms of the difference between nuclear actin and cytoplasmic actin, does rickettsia have a different motility in those two spaces, or?
All I know so far is that they do it a lot more in the nucleus. So in the cytosol, it's 20% of the bacterium are moving at any time. When they're near the nucleus trying to get in, all of them are making actin tails. And when they're inside that nucleus, they're all swimming around because nearly all of them have actin tails. So I do think there is something different about those compartments. Whether it's, I have lots of theories. Some of it could be, we know that they need profilin actin, and I believe there might be differential amounts of active profilin in the nucleus versus the cytosol. It's possible that some cells actually might have different levels of PPIA in the nucleus versus the cytosol. That's something I just recently stumbled upon. So I don't know in our system if that's happening just yet, but I think you're right. There's got to be something different between these two spaces. And
also the cytoplasm is a lot more dense in terms of all the materials. So
yeah, I forget. Somebody told me recently what the viscosity of the inside of the nucleus is too, and I cannot remember if it's more or less than the cytosol, but it's an important thing to think about. Like, it's not just viscosity, it's stuff like it's running into. So yeah.
Thanks.
Fascinating talk. Thank you. Several questions come to mind. One is regarding Wolbachia, and see what's the similarity between what you see in rickettsia versus the Wolbachia, which do enormous things for the host cell, right? So they modify so many things. So I don't know whether you've seen similar factors that are now exported out, whether or not similar things exist in Wolbachia.
Yeah, the effectors that we found, other than that amidase, Alan has done AlphaFold to Dolly, AlphaFold to Fold-seq, Hammer, HH-Pred, and we just keep hitting, other than they have a coiled or a pentapeptide repeat core, there's really nothing that we've seen that's hitting in Wolbachia specifically. But you're right, it seems like there should be somewhere there's some interesting overlap. We have not done an exhaustive check, and actually there's a postdoc in Ruth Lehman's lab who's doing a lot of Wolbachia work that I should ask her if she's ever tried to compare and contrast identical proteomes. Yeah.
Another question was about bioenergetics and metabolism overall. When these bacteria or rickettsia actually are inside this double membrane, in which you said there are no nuclear pores. So how are they acquiring things that they need to grow? Are they having transporters coming over, getting in there, or
What I don't know is how, I mean, it's possible, right? Like they could insert some, we don't know the identity of the proteome of these protrusions as they're pushing in that space. We also don't know how much of that back end is sealed, because there could still be stuff flowing in behind them to help them push against there and also get metabolites. But eventually they break out of that and get in the nucleus. So then the nuclear pore complexes
But you're
can't get
getting
stuff.
actually division occurring within those double membrane structure, right?
What was that?
We're getting division of bacteria, we're dividing.
Yes, they are definitely dividing in that
So
space
they are
too.
getting some nutrients in there, right? So,
I would assume so, yeah.
yeah. Other
We haven't done something like if we, flowing in a dextran just to see if it can keep getting into, or doing some kind of FRAP experiments might be nice to see how much access is coming into that protrusion.
Do you get any DNA damage occurring at all of the nuclear DNA?
So we are trying to look into that. I think Hannah tried to do tunnel assays and they were not showing any obvious DNA damage there, but I think you need a lot of DNA damage for that to get picked up. So there are some, I think, other markers we're looking into to see if we can see early stage DNA damage. But I mean, it feels like it should. There's a lot of stuff happening inside there, but it's interesting to think, again, how the cell is not responding to that by inducing apoptosis and clearing out this infection. So it seems like the pathogen is limiting the effects of that.
Really beautiful talk. Lots of questions, but I'm going to limit it. So I was just curious, your phenotype with the rough ER, like wrapping around the bacteria, was so fascinating.
Mhm.
Does it always stay as like kind of like this like double membrane, or does it ever like fuse, and are the bacteria ever inside the
They
ER?
can get out. So,
Okay.
so Caroline's very recently been doing these extremely long movies to kind of watch when they form them and then how long and how persistent they are. Because when our initial story came out, we had watched maybe for an hour and a half, and they stayed in this interaction. But she sees them kind of, there seems to be some characteristic time, maybe 8 to 16 hours post-infection, that they can start to form these. They sort of expand in terms of intensity of that signal on the confocal, and then they start to break out, and I forget what that time point is. So I don't think they officially seal and stay, and we've never seen that completely wrap in the EM and seal around them, because again, they're getting out, so.
And
It's weird.
is that happening in tick cells too?
There is a single report where they noted something that looks exactly like ours. And so we are in the process of trying to test this in tick cells. Tick cells are not easy to work with. And so it's been really hard to get like ER stains to work in there. So we've been trying to get some ER markers. We just probably should do some EM, honestly, and test it, but that's on the docket. Yeah.
And then I guess, so you know that I love cell death, thinking about apoptosis. So that was fascinating that, right, SRFE, right,
Yeah.
somehow or somehow interfering with apoptosis. Do you have any thoughts on what's happening there?
We
I
have,
was
so we've been
thinking about-
toying, we've been toying around with this idea of like nuclear repair and trying to figure out exactly what it's targeting. That's one of the things that Hannah's been looking at. There's also potentially some lamin, very cryptic lamin binding sites. And so we don't know if it's sort of buffering against those repair processes and maybe facilitating them. And without them, I suppose then the cell would start to trigger its own sensing of nuclear disruption and then cell death on that process. I think she's in the process of also treating with different inhibitors to see if she can block it in the absence of SIRFE to see if she can rescue that phenotype. But yeah, I mean, we're diving now into nuclear envelope biology in a way that I didn't expect to be. So learning a lot there too as well.
Yeah, and so I guess I'll ask one last question, sorry. So yeah, so I think about nuclear integrity breakdown a lot, like I collaborate with people who study this in eukaryotic cells. And when the nuclear envelope breaks down, that's when you get cGAS-STING signaling in response to like endogenous like DNA.
Yeah.
So do you know if that's happening?
We haven't looked
or-
at that. We should do that. Yeah, I like that.
Yeah, that'd be cool.
Yeah, thank you.
Okay, thank you all so much. Thank you to the trainees for their talks and to Rebecca for her wonderful talk. Before we break for reception and dinner, Vincent just has some words to say.
Sorry to be between you and reception. So thank you again for the wonderful talks, the trainees as well. This is our last event of the academic year, and this is our election year. So I am pleased to announce the results. Kyle Rodino from Penn, he will be the president. Jason Heino from Rowan, he will be the president-elect. Pina Frederico will continue to be our secretary. And because we have vacancy, Simon Knights will be our interim treasurer. And that also means I'm stepping down. And so I want to thank everyone within the branch and the executive committee for your support for the last 2 years. And I look forward to what Kyle and Jason will do in the next 2 years. So thank you very much. Enjoy your reception. Thank you.